Here is your question. What is ChIP-Seq? Is it A chocolate chip brand? B. A potato chip brand? C. A type of computer chip? Or D. Chromatin immunoprecipitation sequencing?
Chromatin immunoprecipitation sequencing. What do these words mean to you? You might be thinking of DNA, the immune system, rain, or most likely solutes and solutions, and lists of some sorts. These are all great, but let's visit some important concepts and come back to it.
Keep in mind, in every cell there's a nucleus containing chromosomes, and that is made out of chromatin, which is in turn made of DNA and histones, the packaging proteins. Also remember, DNA can be bound to countless other proteins during the many biological processes of a cell. These proteins, such as transcription factors, chromatin-modifying enzymes, and modified histones interacting with DNA are all important for gene regulation and expression in eukaryotes.
So how can we determine where these proteins bind to our DNA? This is where ChIP sequencing comes in. Its main purpose is to identify the locations in the genome bound by proteins. Here are the steps. The first step is gluing the DNA and proteins together. This is where we use a solvent, like formaldehyde, to create covalent crosslinks between the DNA and proteins. This makes sure they hold on tight. Step 2 is cutting the protein-bound DNA into smaller fragments of about 300 base pairs. This is done through the process of sonication, which uses sound energy to agitate particles within the DNA and fragment it. Next, we introduce antibodies attached to insoluble magnetic beads. What is an antibody? It is a large Y-shaped protein usually produced by B cells that are involved in our immune system. There is a region at the tip where specific molecules, antigens, can bind. Antibodies are specific for a particular antigen. In the case of ChIP sequencing, the antigen is the protein we're interested in.
The next step is immunoprecipitation. Here we use a magnet to isolate all of the antibound proteins which we're interested in. We then wash away and digest the rest of the fragments and proteins, including histones. We then warm our sample. This basically reverses the gluing step and strips the DNA of all the bound proteins, including the associated histones. Once everything has been warmed up, we purify the DNA through various washes to obtain our target DNA sequence. The DNA is then sequenced using a genome sequencer. To amplify the data, a polymerase chain reaction, also known as PCR, is used. One run of sequencing can scan the entire genome with high resolution and determine the precise locations of where the protein of interest binds to the DNA. When sequenced, the fragments are mapped, and regions which have a high concentration of tags are also identified. These regions correspond to the binding sites of protein to DNA.
When would you want to use ChIP sequencing? Scientists can use it to precisely identify all the DNA segments in the genome that are physically associated with a specific DNA binding protein. Pros of ChIP sequencing. ChIP sequencing is unbiased. Other techniques are biased because they require probes derived from known sequences. ChIP sequencing does not require prior knowledge. Also, ChIP sequencing is universal. You can study the entire genome of any organism. Cons of ChIP sequencing. It's time-consuming. Antibody development and quality affects the success rate of ChIP sequencing, but testing every antibody takes time. It's also expensive. ChIP sequencing requires a lot of tissue, which might be an issue if using rare sample types. Now that we know what ChIP sequencing is, let's talk about a real-life application. ChIP sequencing has been used to identify important interactions between chromatin and estrogen receptor binding sites. Estrogen is a steroid sex hormone that directly targets DNA and affects gene expression, leading to multiple changes in the body. Our growing understanding of such links helps us discover the molecular mechanisms and bases of disease.
So now that we've explained ChIP-Seq, let's go back to the beginning and revisit the definition of the name: chromatin immunoprecipitation sequencing. We now know that this has nothing to do with the rain on a gloomy day, but in fact, it allows us to determine the exact location of where specific proteins bind to DNA. Remember, this is super important for understanding various factors that affect gene regulation and expression, something that is so important to the field of science. Maybe I should go back to focusing on winning $1 million.
I picked D, chromatin immunoprecipitation sequencing.